Hey, Bio30s, it's Manchak here, recording your video lesson for concepts C4 and C5. I wanted to, I'm going to record this one without any video, just because I haven't been happy with the sound that I was getting on the videos before, and I want you guys to have the opportunity to see some writing on this one because I'm going to have to work my way through a few things with you using some writing tools. So I hope that works okay for you. Let me know if you do want to see my pretty face again, and I can put that back up, and it probably will come back up on subsequent ones, but I wanted to do this one first. So we are moving into concepts C4 and C5, and based on the concept checklist that you can see here. These concepts are going to be C4 is to describe the evidence for dominance, segregation, and the independent assortment of genes on different chromosomes as investigated by Mendel. So with that said, this is where we are transitioning now into classical genetics, and it's called classical genetics because it had all of its work started by somebody by the name of Gregor Mendel, whom I'm going to introduce you to shortly. So we're going to do a little bit of a background. Concept C4 is a little background on all of this stuff, and then we are going to get into actually trying to figure out what we call patterns of inheritance. And we're going to play around with different kinds of patterns of inheritance and allow us to make some predictions about probabilities of certain traits showing up on certain individuals if we know what their parents' genetic code might look like. So we're going to play around with this a little bit, experiment a little bit, and see how it goes. I'm going to warn you right now, concept C4 is going to be a relatively reasonably short concept, shouldn't take us too long to get through. Concept C5 is a long concept. So here's what I'm going to do. I'm actually going to record 3 videos. This video that you're watching right now is the introduction to the classical genetics stuff, and it will cover concept C4. And then I will do 2 videos for concept C5, one that will do the basics of what we call sort of monohybrid crosses and understanding a Punnett square, which you guys saw back in Science 9. And then I'll do a second video that'll go a little bit deeper into these things like incomplete dominance and codominance, multiple alleles, dihybrid crosses, stuff like that. So that's the scoop on that. So you're going to end up getting 3 videos for this section just to keep things manageable for me and for you. And then what I'll do, as always, is I'll tack on some practice stuff for you at the end of each of the videos, just so that you're aware of things that you can be looking at in those videos to reinforce what you're learning. So for concept C4, which we're doing right now, these are the notes I made for myself, but it's probably good for me to share them with you too. You're going to want to watch out for the terms that we're using. Particularly, we're going to start out talking about alleles versus genes and trying to understand what those are. Then I need you to understand, and that'll link back into chromosomes again, which we talked about in C1 through C3. Then dominant versus recessive will come up. Genotype versus phenotype, homozygous versus heterozygous. All 3 of those terms are going to come up in those videos. I don't have a resource video for you in this section. We'll get into a bunch of others moving forward, and there will be some practice work for you, just a set of questions on page 600 of your textbook. So we'll get to that in the video. So I'm going to just switch over to our PowerPoint and get started on this, if you guys don't mind. So we'll get rolling on that. So this is our introduction to early genetics stuff, and getting into talking about what we call classical genetics. And all of this owes its origin story to Gregor Mendel. And Gregor Mendel was the individual who really proposed a lot of what's going on with our understanding of how genetic information is passed from one generation to the next. So let's get into talking about that. I'm going to rewind a little bit and just set the stage. So biological traits are controlled by genes located on chromosomes that are found in every cell of the body. So if you can imagine, over here is a chromosome, and that chromosome is a tightly wound up collection of DNA. And in the DNA are specific sections that represent genes. Okay, one gene basically in general controls the manufacture or production of a particular protein. And it is the sequence of the individual, what we'll learn about in the next section of Unit C, the individual base codes that codes for this specific protein that we're looking at. Okay, and inside of these genes and inside of these chromosomes are the genetic information that gets passed from one generation to the next. So genetic traits occur because of how the mother and father's genes on homologous chromosomes work together with each other. So remember in the last section, in concept C2 and C3 and C1 too, we talked about how genes are organized, or sorry, chromosomes are organized into homologous pairs. And a homologous pair means that the chromosome is basically identical, except that with the same genes on it, except that one came from your mother and one came from your father. And how those two chromosomes work together in that homologous pair decides what traits you end up with. And all of this is related back to what is passed down in your chromosomes from your mother and your father. This is basically what we call heredity, the passing on of genetic information from parents to offspring. So, with all of that said, I want to just talk about the term allele with you here, because that's what works together with this idea of homologous pairs and your parents' genetic information being passed down from them to you, and how those homologous pairs work together. So let's imagine here that we have a pair of homologous chromosomes. And for argument's sake, let's say that this chromosome came from the mother. We're talking about flowers now, so the mother plant, and this one came from the father plant. Okay, this homologous pair is genetically more or less identical. It's got the same genes all along this chromosome. So, for example, here is the gene that determines flower color, and that occurs at the same location on each chromosome. Okay, so this is what we call the locus for flower color. Locus means location, or the spot on the chromosome where we find that particular gene. And we have two possibilities in this flower. Flowers can be purple or flowers can be white. And if this came from the mother flower and this came from the father flower, then they have the same gene, right? It is the flower color gene, but they have two different alleles. One allele codes for purple flower color, and the other allele codes for white flower color. And it's how these two genes, the one that came from the mother and the one that came from the father, it's how they work together that determines whether the individual that has these chromosomes has purple flowers or white flowers. Now, before we can determine all of that, I want to sort of go into a little bit more background before we can get a little further with that. So let's talk then about Gregor Mendel. So keep that idea of alleles in the back of your mind a little bit, because it's going to help us to understand what Gregor Mendel did. Gregor Mendel was an Austrian monk who gave us our first understanding of inheritance in about the 1800s. And I want to pause at this point and just point out to you that Gregor Mendel did this work long before powerful microscopes were in play. Yes, there were microscopes around. Long before we had an understanding that it was actually DNA that was the mode of inheritance that passed genetic information from one organism to another. He really didn't have an idea about that. But what he did is he just studied pea plants, literally garden pea plants. And he looked at the transmission of traits in these pea plants, and ultimately he laid the foundation for the science of genetics. He basically took those pea plants and cross-pollinated them, or created different hybrids from those by just cross-pollinating them. So he used garden pea plants to do this, and it was very, very simple. And because he was an Austrian monk, he didn't leave the monastery very often, and he had time to work with these plants and play around and write about his findings and get them down for us to see. In his experiments, he used garden peas because they had a series of traits with only two options. For example, the peas themselves that came out of the pods that were grown by the plants could be wrinkled or smooth. In the shape of the peas, the appearance of the peas, or the color could be green versus yellow for the pods that were created. And so he had lots of options. And the other thing that was great about them is that they self-pollinated. So the traits were apparent in all plants he used, not just some. And it allowed him to play around with pollinating them as well. So Gregor Mendel sort of kicked off all of this in his experiments. And what he did, we'll get to later on, is he came up with a series of rules that we can use to describe genetic traits and how they are inherited or passed down from one generation to the next. And it links up with probability, the ability for us to predict what an offspring might look like if we know what the parents look like. So let's circle back and provide a definition then for an allele, because this is going to become extremely important as we move forward. An allele is one of two or possibly more alternative forms of a gene. So for example, a smooth allele, smooth shape to the pea itself, or a wrinkled shape to the pea. And so there's two alleles. One form creates the smooth, the other form creates the wrinkled. Okay. And so they're alternate forms of the gene. That's the way I kind of like to think of it. It's still the same gene. That would be the appearance or feel of the pea itself, but there's two alternate forms. It could come out smooth or it could come out wrinkled. In humans, as with most other multicellular organisms, chromosomes come in pairs. One chromosome from the mother and one from the father. And on each of these chromosomes are genes, which constitute your mother's and your father's genes. These genes are broken into two alleles for now for us at this point. And so that means that your father will supply an allele and your mother will supply an allele. And how those two alleles work together is what determines your traits. So if you have two of the same allele coming together, then you're going to end up with a particular trait. If you have two different alleles, you may or may not end up with a particular trait depending on how things work out. So we'll explain to that and get into it in a little bit here. What Mendel discovered as he worked with his pea plants was that you could determine that an organism's traits fell generally into two categories, what we would call a dominant or a recessive trait. A dominant trait is characterized by is a characteristic that is expressed when one or both alleles in an individual is in the dominant form. And dominant alleles, when they are present, mask the expression of the recessive allele. So let me just go back up here for a second. Let's say, for example, that the purple color was dominant over the white color. That would mean that if this individual came together, okay, this dominant allele would overrule this recessive allele, and these two chromosomes would work together to make a purple flower because of that. I hope that makes sense. Because this is dominant, okay, we're saying purple is dominant, that overrules the recessive allele. So the only way that we're going to have white flowers then is if we get an individual that has two of these white alleles. One white allele that came from the mother and one white allele that came from the father. Hope that makes sense for you. So then talking about a recessive trait, it's a characteristic that is expressed only when both alleles in an individual are in the recessive form. So we got to have that both alleles bit going on there. So to discover his principles of dominance, and that's what we just talked about, is that alleles have dominant versus recessive abilities. Mendel crossed pea plants that were round and smooth with wrinkle-seeded plants. Sorry, I had to have a sip of water there. My voice is getting scratchy. And what he did is, so basically just imagine this, is Mendel took a plant that was had round seeds, smooth seeds, and he crossed them with wrinkled seeded plants. And what he found is that all of the resulting offspring from that first generation, what we call the F1, the offspring or the F1 generation, they all had round seeds. So just pausing there and not going any further, I can conclude at this point that round seeds are dominant over wrinkled seeds, because if he took a round plant and a wrinkled plant and he brought them together, and he did it a bunch of times, and every single offspring came out with round seeds, we can conclude that round seeds are dominant over wrinkled seeds. Okay? Based on what he observed along with other testing, Mendel realized that some traits or alleles dominated others. In other words, they always produced a particular characteristic in offspring. Dominant genes, a certain characteristic like in dominant genes, and some traits never showed up in offspring with the presence of the dominant gene around. So what I mean by that, or what I wanted to do here, is I want to point out this capital R here and this lowercase r here. When we are representing dominant and recessive alleles, we break everything down to just representing a letter. Okay, and if we use a capital R, for example, capital letter will represent dominant. Okay, so D-O-M for dominant. Apologies for the horrible writing. And a lowercase r will represent recessive. Okay, so REC for recessive. We're going to use this when we start to write down our proof for how certain generations turn out and what the offsprings might look like. So the F1 offspring, which were all round, were then crossed with each other. And what do we expect the offspring to look like? So I'll do a Punnett square of this in a few minutes here and sort of show you an easy way to sort this out. But one of the ways you can look at it is the F1 offspring. So remember, the F1 offspring were created by taking round plants and crossing them with wrinkled plants. Okay, and all of those offspring were round. And then what Mendel did is he took all of those offspring that were created from the initial cross and he crossed them. And he was unsure what he would end up with. And what he found, which was really interesting and important for us to note, is that 75% of the offspring were round, but not all of them. 25% were actually wrinkled. So what he hypothesized is that the wrinkled seeds must have two recessive alleles, whereas the dominant must have one dominant, or sorry, the round seeds must have one dominant and one recessive allele, a big R and a little r. And so that whole experiment was super important in trying to understand what was going on. I will get into this a little more deeply in a little bit, but for now I just want you to get that as Mendel worked his way through this, he started to come up with the idea that certain alleles were dominant over others. And he could prove that by doing crosses of certain individuals and looking and seeing what their offspring turned out to be. This leads us into talking about a couple of terms that are going to come up throughout the course of this section. So C4 through C8 is the section I'm talking about. Please make sure that you learn your terminology here. Understand the difference between a gene and allele and be able to explain it. Understand the difference between a genotype and a phenotype and be able to explain it. Understand the difference between homozygous and heterozygous. So a genotype is the genetic complement of an organism. So I think of the geno part of this as the genes. What are the genes that the individual has? In particular, what are the alleles that they carry? Okay, so what are the alleles that the individual carries? Okay, so that's what we should be thinking about when we think about genotype. So is the individual carrying a dominant allele and a dominant allele? Are they carrying a dominant allele and a recessive allele? Or are they carrying two recessive alleles? Remember, capital means dominant, lowercase means recessive. Okay, that's what a genotype is. These are the genotypes. And it'll get a little more complicated when we move into subsequent concepts. But for now, we'll leave it at this. Phenotype, then, I like to think of as the physical expression. So the pH makes me think of physical expression, the observable characteristics, how it physically appears when a genotype is expressed based on the genotype they possess. So, for example, if round is dominant over wrinkled, so capital R, little r, then an individual that is round could have two possible genotypes, right? Round is your phenotype, right? This is your phenotype. I'll just put pH for phenotype. And these are your genotypes. Okay, so that is a genotype, and that is a genotype. Okay, I'm just going to put G for genotype. So then an individual that is round for its phenotype could have two possible genotypes, right? Because this is going to produce round-seeded individuals, and also this because the big R overrules the little r in this case. Okay, an individual that is wrinkled, again, this is a phenotype, will only have one possible genotype. They could only be little r, little r. So I hope that distinction makes sense to you. What a genotype and a phenotype is. Very, very important terms for us as we move forward. The next thing that we can do is categorize things and become more accurate with how we're representing things. So, homozygous individual is an individual where both alleles are the same. Okay, so homo, that prefix there, means same. Okay, so if we have an individual who is big R, or an individual who is little r, those individuals are homozygous. In other words, their alleles are identical. If both alleles are recessive, like this individual here, they will be considered homozygous recessive. Okay, if both alleles are the dominant allele, like in this individual here, we would call them homozygous dominant. An individual is said to be heterozygous. Hetero means different. Okay, fairly simply, just put it as different. That is horrible writing. So a heterozygous individual means that they have one of each allele, one dominant and one recessive allele, like that individual. And we do not make the distinction calling them heterozygous dominant or recessive, right? We just say they are heterozygous. Hope that makes sense for you. So what I need to talk about now then, I'm just skipping ahead to remind myself of a couple of things. What I need to talk about now is the idea of segregation. Okay, so Mendel came up with this first law where he said that individuals are or express based on whether they have a dominant allele or not. If you have a dominant allele, you express the dominant trait. If you do not have any dominant alleles, in other words, you just have recessive alleles, then you will express the recessive trait. So that would be what we'd call Mendel's law of dominance. What Mendel also came up with was an idea called segregation, or the law of segregation. So after concluding his experiments, Mendel also postulated that the two copies of a gene in a pair undergo segregation during the formation of sex cells. So when the mother and the father are creating their sex cells, the alleles are separated from each other in order to create an egg or a sperm that has only one allele in it. So if the original individual was big R, right, then they are going to produce gametes that have only big R alleles in them. I hope that makes sense. So when we start out with an individual who is big R, and they're creating gametes, right? These are going to get separated from each other in meiosis 1. and the cells are all going to end up with big R alleles in them. If an individual is big R, little r, and that's what we're talking about right here, after meiosis 1, those alleles get separated from each other. And you can get some gametes that have the big R allele and some gametes that have the little r allele. So this idea of segregation means that when we actually start to work with a Punnett square in the next concept, we'll actually be separating these out, assuming that segregation takes place and that we're going to have individuals that have only big R gametes and individuals that could have a big R or a little r gamete that is being put forward. Then when the gametes combine during sexual reproduction, the dominant and recessive alleles are again paired with each other. So if these are two separate individuals, then they might each provide a gamete. And if it was this gamete from this individual and this gamete from this individual, then they would produce offspring that would only be big R, little r. Okay, I hope that makes sense to you. So then this is the process of fertilization here that produces a new individual. So Mendel's laws of heredity that I want to talk about with you. These three I need you to know, and I need you to memorize them and understand what we mean by each of them. So the first law is what we just talked about, what we call the law of segregation, is that each gamete gets one of the pair of alleles from the mother or from the individual that they're the individuals that make them. So if you have that homologous pair, they are going to either get the mother's allele or the father's allele, not both, moving on to the next generation. And then that's going to combine with another individual through fertilization to make the offspring there. The other one, and the law of dominance is sort of what we just talked about back up here. And the law of dominance is basically that if the dominant allele is present, then it will be expressed physically. If the dominant allele is not present, then the recessive alleles are the only ones around. They will be expressed. The other one is the law of independent assortment that we really haven't talked about, but I'll try to explain it as best I can right now. Characteristics are controlled by genes that occur in pairs, one from each parent, and each pair of alleles segregates independently of the other pairs of alleles during gamete formation. What this means, and I talked about this in concept C2 with you, is when the first division of meiosis is taking place and we're lining up all of the homologous pairs along the metaphase plate in meiosis 1, so metaphase 1, if we have your mother's allele or mother's chromosome on the left side and your father's chromosome on the right side for our first pair of chromosomes, that doesn't mean that we have to have the mothers on the left and the fathers on the right in the second pair. Right? It could end up being that fathers is on the left and mothers is on the right. Or it's just as likely that mothers is on the left and fathers is on the right. And even though we've had, let's say, mothers end up on the left twice in the first two pairs, it doesn't mean that mothers will be on the left in the next pair. It also doesn't mean that fathers has to be on the left in the next pair. It is what happened in the first pair, and all subsequent pairs is independent of what happened previously. So it's just how they line up along the way there during my metaphase 1 of meiosis 1. If that doesn't make sense to you, fire me an email and I can do up a short video that might explain it a little bit better than it does right now. So that is the end of concept C4 and what I have to cover. Have a quick look at page 600 and the questions 1 through 5 on there, just as an idea of what I think is important from this concept. If any of this doesn't make sense, please fire me an email and let me know so I can get back to you with an answer either in a video form or some other sort of form. Just remember for concept C4, I really want you to understand the terminology here. So if you walk away with anything, spend the time learning what a gene is, and what an allele is and how they are similar and different. Learn about what dominant versus recessive means and how we represent dominant with a capital letter, recessive with a lowercase letter. Learn the difference between genotype and phenotype. Learn the difference between homozygous and heterozygous. And then take the time to learn about the three laws of segregation, independent assortment, and dominance. And then that should be enough for concept C4.